Your gut bacteria, they're super cool, but the reality is your gut bacteria and probiotics, they are not a magic button, a magic remote control for your body. You can't simply press a button and you're skinny, or press a button and you're happy. And certainly there's no button that's going to suddenly reverse autism. There's a great deal of hype around the microbiome. And if someone is selling you that button, then I would say congratulations, you have met the wellness industrial complex, wearing their lab coats, probably bought on Amazon, but really not selling you real medicine. But the microbiome is also not fake when it comes to health. I mean, some gut bacteria can be used to help cure a deadly infection, and some of these bacteria can really mess with your drugs, and some of these bacteria are involved in the process of risk for cancer and tumorigenesis. So microbiomes are important for health. It was one of those situations where the science is both really, really interesting and really, really annoying at the exact same time. Hello, I'm Cecil Merlin Lewis, Dr. Merlin for short, and I've been studying the microbiome for nearly 20 years now, and I want to talk to you about where is it overhyped and where is it really, really relevant for health. And we're going to start with the really, really relevant. So let's dig in. Let's put this into three piles. Let's have the real medicine pile. Let's have the probably important but complicated pile. And then let's have the please stop selling fairy dust pile. All right? The first pile is the stuff where the microbiome is real health. This is where the microbiome walks into the room, flips the table and says, I'm the cause, or I'm the remedy for this particular thing that you're going through. The best example that I can think of has to be C. diff infections. And C. diff is a nasty gut infection. I'm talking about horrible diarrhea that comes back and back again, can be resistant to antibiotics because you use antibiotics to wipe out the microbial ecosystem, but that ecosystem is also part of the players in keeping C. diff at bay. Now your entire ecosystem is just a mess, and antibiotics aren't helping, and C. diff is getting worse and worse, and it could be life-threatening. People can die from this. It's absolutely terrible. But there's a way out of it, and it's through your butt. So in some cases, if antibiotics has failed to eliminate C. diff from your rejuvenated ecosystem, then what if we change that ecosystem entirely through a transplant of some kind? What if we do a fecal microbiota transplant, which is highly screened medical process stool that is being used to reinvigorate the ecosystem? And what's great is these fecal microbiota transplants, they actually work. This is one of the strongest success stories in microbiome medicine. There are even FDA-approved microbiome-based therapies now. Some of these meds you put in your butt, some of these meds you put in your mouth, and they're basically bacterial spores that'll come in and rejuvenate that ecosystem. That's quite a bit less invasive than doing a complete transplants. That means that one of the most validated microbiome treatments involves a gut ecosystem repair, a true microbiome manipulation, because that's what a microbiome is. It is a biome ecosystem. That's really cool stuff. Tiny nerd detour here as I help explain microbiome studies if you happen to be Googling or searching them. Older microbiome studies often use 16S RNA gene sequencing, which is creating an inventory of bacteria that are there, and you can compare that inventory to whatever particular health phenomenon that you're interested in. Older microbiome studies were great. We found lots of initial patterns, but even though you know which microbe might be there, you don't really know what all the microbes are capable of. Newer studies often use something called metagenomics. And metagenomics will often sample the entire DNA assortment that is inside of an ecosystem. You not only get who's there, like what bacteria happen to be there, you also get a more complete picture of what that ecosystem is capable of, what the bacteria can actually do, what chemicals they can produce. And you can complement that with other forms of omics, transcriptomics and metabolomics, which looks at the small molecule chemistry. All of these other data sets are more common in studies today, giving you a more complete picture of what the microbiome is capable of. A lot of our initial ideas about the microbiome have really changed because we have new data, more complete pictures of what this ecosystem actually is doing. Enough of the nerd break. Let's get back to microbiomes and health. All right, now I got to say a great big word. This is going to be interesting. The second rock solid area is pharmacomicrobiomics. Oh, I almost got it. Let's try that again. Pharmacomicrobiomics. I think I got it right that time. Pharmacomicrobiomics. Pharmacomicrobiomics. Horrible word, but a really, really important idea. It means your gut bacteria can chemically modify drugs. And I'm talking a lot of the drugs that you take. One big study tested 76 gut bacterial strains against 271 medications and found that microbes could metabolize, they could change, chemically alter about 2/3 of those medications. That's pretty crazy. So think about it. When you take this medication, the chemical of that medication doesn't just meet you, it meets an entire microbial committee living inside your colon, and some of those microbes attack the medication. One famous example, one of these microbes that attacks is Egerthalia lenta. Some of the strains of this particular bacteria can inactivate digoxin, which is a common heart medication that's been around for many years. This bacteria is not evil. It's not purposefully attacking your medication. The medication actually resembles a plant chemical that the bacteria would normally be going after. And so here's an example of where our medications resemble other forms of chemistry, and that ecosystem responds in a particular way because it resembles, well, in this case, a plant chemical. This is where it gets really exciting to me because through understanding the biology and understanding the ecosystem, we can impact people's lives in a very positive way. This isn't like buy a probiotic and it's going to change your life forever. This is why certain medications work well for some people, but not others. It's part of that story, and that's a really important story. Another quick nerd break about Egerthalia lenta. This bacteria has been a bit suspicious for quite some time. It is found in connection with autoimmune diseases, not all autoimmune diseases, but certain autoimmune diseases. In fact, its abundance has been connected, for some individuals, has been connected with the severity of rheumatoid arthritis. This is certainly a bacteria that we want to keep our eyes on and understand its biology even more. Now let's talk about some tiny criminals. Some strains of E. coli carry something called the PKS island. While that sounds like some kind of vacation resort for bacteria, it absolutely is not. This PKS island allows the bacteria to produce colobactin, and colobactin is some bad stuff. This is a toxin that can directly damage DNA, not metaphorically damage DNA, like actually break the DNA. And the wild part is that scientists can find a DNA fingerprint, a sort of DNA break and repair fingerprint that leads back to that bacteria. And where do they find it? They find it inside the cells of tumors, such as those tumors that you would find in colorectal cancer. So now what's going on here? Well, it seems like the bacteria, the chemical that it produces, is leading to damaged DNA. And that's not necessarily directly causing the cancer right away, but it's certainly enhancing the risk for cancer, because in the end, when it comes to becoming a cancerous cell, it is about mutations that have gone awry. Very fast nerd break, but I can't help it when we mention E. coli, because every time you hear E. coli in the news, it's like always the bad guy, right? E. coli in lettuce or whatever, causing this horrible outbreak. But E. coli, overwhelmingly, most E. coli are harmless, and some of them are actually quite useful. That's the thing about E. coli that's sort of fascinating. E. coli is highly diverse set of bacteria. In fact, there's only 20% of the E. coli genome is considered core, essentially universal among E. coli. The rest of it is very plastic. It can be quite different among E. coli. And so E. coli have been fascinating, often, completely harmless, some of them harmful, but some of them beneficial. And because of their biological properties, they have been not only useful for understanding basic biology, but they've also been very important parts of the laboratory in certain biomedical sciences. Let's enter that middle zone where the microbiome is clearly important, but very, very easy to overstate. Cancer immunity is a really good example. There are studies showing that the gut microbiome can influence how well some patients respond to immune checkpoint inhibitors. These are drugs that help the immune system attack cancer. So, in some small trials, fecal transplants from responders have helped the non responders to once again respond to the immunotherapy. I know that was a bit confusing, but I hope you caught it because that's genuinely exciting that you can make a non response to a cancer treatment a response to a cancer treatment through a fecal transplant. Here's the thing about antibiotics and immunotherapies, and the situation is pretty complicated. For many cancer treatments, an antibiotic step can be absolutely life-saving when that cancer treatment suppresses the immune system, leading that individual to be very susceptible to invasive and pathogenic bacteria. But the downside is that antibiotic exposure near the time that somebody is undergoing immunotherapy has also been associated with worse responses for some individuals in some studies. You can see this gets pretty messy. This is an area that needs a lot more attention, and yet it's an area where the findings so far are already really highlighting a value of the microbiome. Let's talk about early life, because this is actually one of my favorite ones personally. I'm not too sure why I'm so excited about this one, but I really like this a lot. When it comes to newborns and babies, the first 1,000 days of life is a major window for their immune system development, how they're going to fight off diseases, infections in the future, their immune system. And we've learned some really cool stuff in its connection to the microbiome. This idea, which is pretty well supported now, that your immune system is programmed by your microbiome before you're even a toddler. Kids whose guts are disrupted by C-sections, for example, or by early antibiotics, have measurably higher risk for allergies and asthma later on in life, like by age 6, 7, or 8. It's a really critical window, this first 1,000 days, quite clearly. This is certainly not to guilt trip C-sections or antibiotics. C-sections save lives, antibiotics save lives, no question there. But these highly valuable interventions can have some downstream consequences. Fortunately, there are great studies showing that this risk for allergies and asthma can be mitigated, and there's a number of really cool ways that can happen. One cool example is if you happen to have a toddler already in the house, somebody who's relatively close in age to your newborn, they can serve as a way to inoculate that newborn just from playtime and gross, sticky hands. Babies that have siblings that are close to age in the house tend to not suffer from the allergies and asthma at the same risk levels as individuals who lack that in the household. Now, this is not to say that that's the only way to fix this issue with allergies and asthma, but it is one example of how the ecosystem of our home can be impactful in shaping your microbiome, but also shaping your risk for allergies, eczema, and asthma. You might be saying, I have allergies and I have eczema and I was not born by C-section and I had plenty of siblings. Remember, one of the things that gets very confusing when we talk about this complexity of the science is we're talking about risk factors. Yes, there's always individuals that do not fit that mold, but if you're looking over a large number of individuals, you're going to see very strong trends. We're often talking about your risk for allergy, not that you can never have it if this happens. That's just not the way complex diseases work. And the vast majority of diseases are sadly complex. They're not simple. But that doesn't mean we don't care about those risk factors, because the more we can reduce those risk factors, the less misery there's going to be in the world. They're important, even though they're not straightforward. Continuing on this story about complex diseases, we can talk about chronic diseases in the microbiome. I'm talking about diabetes, inflammatory bowel disease, liver disease, depression. You might have seen news articles about these topics in the microbiome because they're relatively popular. And researchers often see a particular pattern where helpful bacteria are going up and unhelpful bacteria are going down in association with these particular complex diseases. But As you might predict, the story is not so simple. One key word in explaining some of this is butyrate. Butyrate is a small chemical. It's a short chain fatty acid. It is a chemical that is broken down when your body digests fiber. You've probably heard fiber is really good. One of the reasons why fiber is really good in your diet is that your bacteria, your gut bacteria, will break it down into butyrate. And butyrate really is some good stuff. Butyrate is fuel for your cells. It also helps support your gut lining. Supporting your gut lining is pretty important, as you can imagine. I mean, if you think about your intestines, you want the stuff inside the intestines to stay inside the intestines until you're ready to remove them on your own. You do not want that stuff crossing your intestinal lining. So having a nice, strong intestinal lining, yeah, that's pretty valuable. Butyrate, really, really good stuff. When you hear studies that say this particular chronic disease involved the loss of butyrate producing bacteria, you might think this is bad stuff. Like now your gut's maintenance crew is definitely understaffed. But here's the trap. If the maintenance crew is missing from the building and the building is on fire, you certainly wouldn't assume that the maintenance crew was involved in the fire. The maintenance crew could obviously be impacted by the fire completely independently. The fire here, the fire in this terrible metaphor, is actually inflammation, an inflamed gut. You know, when you get inflammation, your eyes are itchy, your eyes get all swollen, inflammation. That inflammation is happening also at times inside your gut. And that inflammation, unsurprisingly, has a big impact on the ecosystem inside of your gut. It changes the oxygen levels, for example. It changes the mucus inside the gut. It changes the immune activity. And so as a consequence of this, it changes what bacteria can be successful in that environment, including those butyrate producing bacteria. While those bacteria can be the cause of some of these complex diseases or a player in that cause, they may also be the casualties, and maybe a little bit of both. The science here is a bit fun in its complexity, but also a bit frustrating in its complexity when you're really just trying to impact people's lives. And a good example of that would be diabetes. For years, researchers have seen microbiome patterns in people with type 2 diabetes. I found these early studies so exciting. I still find them very interesting, but there has been some realizations in how we were doing those studies in the past and what's really at play. For example, microbiome researchers found that a drug that's commonly used to treat diabetes, metformin, can directly impact the microbiome. So it changes the microbiome. In this case, the microbiome is changing not in response to diabetes, but it's changing in response to the drug that was given to somebody because they had diabetes. And again, this is not to say that there isn't some complex relationship between the microbiome and diabetes, but it is showing that as we've done these studies, we're understanding layers and layers of complexity. If I think of a metaphor to describe the microbiome's relationship with these complex diseases, that it's not a single suspect and a single set of fingerprints, it's an entire crowded bus of individuals and everybody's touching all the evidence. Yeah, that's a terrible metaphor. Are they touching the evidence? What are they doing? They're touching the evidence, and some of that evidence is probably important. Yeah, that's fine. Now's the part where I create emotion by getting directly closer to the camera. Where are the areas where claims about the microbiome just ran into a wall? Let's dig in. The first has got to be the claims that the gut microbiome cause autism. The simple answer, no. There are real GI issues among autistic people, and that matters a lot. Those symptoms really deserve care. But the idea that the microbiome can cause core autism traits, that's been massively overclaimed. The major critique, and I'll provide a citation, where a lot of the early studies that were finding a connection between autism and the microbiome leaned too heavily into mouse behavior models that don't map cleanly into humans, and the studies were often too small, and they really poorly controlled for diet. And diet really matters here because many autistic kids have restricted diets due to sensory differences. If you're comparing autistic kids to non-autistic kids, and you find different gut bacteria, you may be seeing autism causing diet differences, which will cause microbiome differences, not gut bacteria causing autism. That's not the right direction. That distinction really, really matters here because bad causality is how you get desperate parents sold on expensive nonsense. That is cruel. Moving away from autism and diet, there are other aspects of a diet and the microbiome that have fascinating patterns but do not seem to have good intervention opportunities for humans. In other words, they're not necessarily something that humans can use and act on. There was a great mouse study that happened in 2013 where if you take a lean mouse and you give it an obese microbiome, that lean mouse will start gaining weight. And that was a really cool study about understanding an aspect of biology. And certainly at that time, you wonder how much that would translate to humans. Well, now it's been another decade or more, and we're discovering it just doesn't translate very well. These lean donor fecal transplants in humans have not produced sustained weight loss, for example. The results are very modest and temporary at best. A related example would be the gut health scores, and whoo boy, are we getting into some dookie here. If your $200 poop test gives you a single gut health score, like it's judging your colon's credit rating, be very skeptical, please. Be very skeptical. Often it's reporting a firmicutes to bacteroides ratio, and they might not be calling it that, but that's probably something like that that's going on underneath the hood of these particular tests. And that ratio has been used in microbiome studies for a very long time, but there's now a current consensus that they're just not useful ratios. They can be greatly impacted on how you do things in the lab. Your DNA extraction procedures, for example, can impact these ratios. So the ratios themselves are not great. Same gut, same person, different lab, different ratio. That is very far from precision medicine. When I think about these commercially available kits, these direct-to-consumer testing of your gut microbiome, they're very cool if you just want to understand bacteria, if you want to understand a gut ecosystem and who's there and start Googling the members of your gut. Man, I love that. Do that. That's fun. That's really, really cool. But don't think that these kits are going to give you real strong practical application for your health or for your diet. The science is just not there yet. There's been a lot of hype. There isn't going to be something that makes it all into an easy package, like I'll look at my microbiome and take this probiotic and bam, I'm cured. I would love that to happen someday. I would love to be proven wrong about that. That would be a wonderful day. But I don't think that's going to happen anytime soon, if ever. I have not exhausted all the different types of microbiome studies that are out there. If there's a particular study that you found fascinating, please put it in the comment section. I would love to take a look at it or comment on it. If you like this kind of content, please subscribe. I'm a new influencer. I am learning a lot about it, and I'm very excited. I hope every week to provide something new and interesting to think about and to talk about. Thank you.